Hello, everyone. It's fantastic to be here. My name is George Murphy. I'm an associate professor in medicine on the medical campus and also one of the co-founders of the Boston University and Boston Medical Center's Center for Regenerative Medicine, which we affectionately refer to as the CREM. So at the CREM, one of our foundational technologies is the use of induced pluripotent stem cells. For you Lord of the Rings fans out there, iPS cells are sort of the one cell that can rule them all, and that you can make all of the cell and tissue types in the body from these master stem cells. At the CREM, we make about 25 different types of cells, many of which you see illustrated here, and you'll also notice each is associated with a particular disease that we might study. More recently, in our studies of aging, we've sort of flipped the script here. So instead of modeling a disease in a dish, we've instead begun to model what Stacy just described, resiliency. So as she noted, one of the most important populations that imbues this tremendous resilience are centenarians and their related offspring. So in collaboration with Tom and Stacy, we built the world's largest iPS library of centenarians and their offspring. We're able to connect this beautiful phenotypic data that Stacy just described to these created lines. We proved that you can actually make iPS cells from people that are extremely old, 110 or better. We plugged this data into biological aging clocks to show that these individuals have a biological age or a molecular age that's much lower than their chronological age. And then finally, what we do as stem cell biologists, we show that we can make an unlimited amount of biomaterial from these master stem cells to study aging and longevity. So how do we go about doing that? How can we study aging, longevity, and resiliency in a dish? So let's walk through our experimental design. So in a nutshell, we take these iPS cells from individuals with exceptional longevity, along with controls with no history of longevity, and we can differentiate them into multiple cell types that are impacted by aging. We then add a series of mechanistically distinct stressors so that we can amplify these signatures, these genes, these transcription factors, these pathways that may allow you to be resistant to disease. And then we profile these signatures both molecularly and with functional assays. So what have we found so far? What's the $20,000 question? And in my last slide, I can describe that. So on the left, when we take neurons, for example, and we add stress to those neurons, in individuals with exceptional longevity, we see upregulation of genes that are related to things like neurogenesis, the birth of new neurons, or genes that imbue resistance or resilience against Alzheimer's disease, for example. In the middle, we see upregulation of one of our favorite pathways. This is protein processing regulation. So these individuals have the ability to look at aberrant proteins and get rid of them out of the queue. And then lastly, as far as functional assays, when we look at the mitochondria from your biology classes, the powerhouses of the cell, we see something that's really unusual. So in contrast to what you'd expect from these really vibrant individuals, when we look at their mitochondria, they actually have much less membrane potential, much less mitochondrial activity than you'd expect. And what's really interesting is that this is in coordination with other long-lived mammals. So if you look at a bowhead whale that can live up to 250 years, it also downregulates mitochondrial energetics and also respiration. So we think this is an imbued longevity mechanism that applies this characteristic. So basically, we think these individuals have a really amazing landscape that we can mine for these interesting variables. And if you're interested in the work we're doing, you can go to our website, www.murphylab.com, to learn more about what we're doing. Thanks.